Cellular respiration is the complete breakdown of glucose, aka sugar. There are two types of cell respiration, but today we're going to be focusing on aerobic cell respiration, and that means that oxygen is used as a reactant. There's three main parts to cellular respiration. The first one is glycolysis, where glucose goes in and we have 2 pyruvate, 2 ATP, and 2 NADH that come out. Okay. The second part is a citric acid cycle, also known as a Krebs cycle. We got 2 pyruvate that go in that one, 2 acetyl coenzyme A, and at the end we have 2 ATP, 8 NADH, 6 carbon dioxide, and 2 FADH. Final step is oxidative phosphorylation. There's two parts to it, which is the electron transport chain and chemiosmosis. At the end of it, we have 26 to 28 ATP. That'll result in a total of 30 to 32 ATP per cellular respiration. NADH causes more production of ATP because it enters the electron transport chain first. There are 3 complexes in the electron transport chain that pump protons. It's 1, 3, and 4. 1 and 3 pump 4 protons, and number 4 pumps 2 protons.
Hello everyone, I'm going to talk about the cell cycle. The cell cycles contain two stages in the cell cycle that are anaphase and meiosis. The anaphase is the largest stage of the cell because it contains 19% of the cell. And because it's the large, it can break down 3 stages. That's G1, S, and G2. Now, in G1, we have the ATP synthesized, the protein synthesized, and the organelle proliferation. Because in the inner phase, we got the envelope nuclei. The envelope nuclei here, and in the nucleus, we have the DNA, the ribosome, and the protein. That's why we have the ATP synthesized protein and organelle. After that one, we have D0. D0 is the stage that our cell is rested and start working for S phase. The S phase is the most important phase of the cell cycle because it's that place the DNA is duplicates, and for example, you have the original chromosome here, and under the influence of the S stage, the original chromosome is replicated by forming a centromere. When they form a centromere, so is replicated. Once replicated, the individual chromosomes are called chromatid. And after the S phase, we got the G2 stage. The G2 stage is this stage where the cell is prepared for the division. So we have the spindle protein and the RNA are synthesized. After that one, I'm gonna talk about the mitosis. Mitosis is a process that makes sure a cell is ready for division, so the new cells produced are identical to each other. The phases are prophase, the pro-metaphase, the metaphase, the anaphase, and the telophase. In the prophase, the nuclear, in the prophase, they are chromosome condensed. The chromosomes are condensed in the pro-metaphase. So each duplicated chromosome appear as 2 identical sister chromatid joined as their centromere, and in some species all along their arm, the cohesion. After the prophase, we have the pro-metaphase. At the pro-metaphase, the chromosomes are more condensed. That's the difference we notice here, some fragment on the nucleus. a fragment, the chromosomes have become more condensed, and each chromatid of each chromosome now has a kinetic core. It have a kinetic core, a special protein structure of the centromere. After that one, we have the metaphase. The metaphase, here the centromere are now at the opposite pole. The chromosomes have all arrived at the metaphase place, a plane that is equidistant between the spindle two poles that are equidistant. They form a line here. Now at the the anaphase is the shortest stage of mitosis, and they are a cleavage of the two sister chromatid of each pair of chromosome. After that one, we have the telophase. The telophase is this phase where the nuclei appear and the chromosome become less condensed. They become less condensed. So that's how it's characterized, the type of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. After that one, I'm going to talk about the cell signaling event. For the cell signaling event, for that one, we have two types. When in absence of in absence of the signaling, and in the absence of signaling, and when the signal is present. In the absence of the signaling, what is happen in absence is of the growth. Is absence is what? In the absence of the goal ahead the signal, a cell exits the cell cycle and enters G0, a non-divided state, and what happens, the result, we have a prometaphase. So the prometaphase, the cell at this stage now of absence of the signal, a cell in mitosis receive a stop signal when any of its chromosomes are not attached to a spindle fibers when it's in absence. Now when a signal come, we have a division, and if a cell receives a go signal, the cell continues on in the cell cycle, and we have some division of the cell that are metaphase and the anaphase. And what happens at this stage, when all chromosomes are attached now to a spindle fibers from both poles, a go ahead signal allows the cell to proceed into the anaphase. So these are the checkpoints of the signal. This is the checkpoint of the signal, and when they are activating, they all have a response on the cell. So that is what happens when the cell receive a signal, and when the cell does not receive a signal.